talk about factors that are going to affect that blood clot or that thrombus that's been formed. And the first thing we want to talk about is the fibrinolytic system, which involves a protein known as plasminogen. But we need to start at the beginning. And the beginning of this system is something that's called a tissue plasminogen activator. In other words, there's stuff in the tissue which are produced by the same endothelial cells that were damaged in the blood vessel wall that triggered the coagulation cascade. So there's other chemicals we haven't talked about. And one of them is this plasminogen activator. And these plasminogen activators will act on plasminogen, which is a protein circulating around in your blood, and plasminogen then becomes its active form, which is plasmin. And what plasmin does is it acts on the fibrin, that cross-linked firm fibrin molecule mesh, and it's like taking a little scissors to it and it chops it up in little pieces, and you end up with what are known as fibrin degradation products or fibrin split products. Either name is acceptable. So in other words, the thrombin in the coagulation cascade gave us a thrombus, a blood clot. But this fibrinolytic system, fibrin lysis or cut up license, cuts up the license and gives us fibrin degradation products, trying to return us back to having normal plasma. So when we have these fibrin degradation products being produced, the clot is going to break down and this serum escapes. So the difference between plasma and serum is that plasma has all these clotting factors floating around in it. And in serum, we don't have the clotting factors because they've already been used up. But if serum escapes on the outside of your body, then you have those yellow drops, which you thought were plasma, when in reality they were serum. But inside your body, the serum escapes and it becomes part of the plasma again. So like within a nanosecond, it's actually back to plasma again. So what's really cool is now we have created synthetic tissue plasminogen activators, and we can give those to people, for instance, who have a clot, which is either causing a heart attack, a myocardial infarction, or a stroke. And this promotes that clot to dissolve and break back down, thereby reestablishing blood flow to that area of the heart or the brain and trying to prevent further damage or limiting the scope of the damage at least. So here on the left side, we have the coagulation cascade, and in the bottom right, we have the green fibrinolysis system put in there as well. So go ahead, take a moment and see how these interact before you continue on. All right, so another system to talk about that has to do with prevention of clotting is something called protein C. So this starts with thrombin once again. So once thrombin is produced, it is going to interact with another protein which is on that exposed, damaged endothelial cell, and it gives us this complex which activates a protein known as protein C. And protein C acts on two of the activated factors in the coagulation cascade. So if we look at this picture on the left, we have coagulation cascade, and they're representing both the intrinsic and extrinsic together there, and this is the final common pathway where we have the thrombin giving us fibrin. So over here, the thrombin, at the same time it triggers fibrinogen to form fibrin, it's also triggering the formation of protein C. And what protein C does is it goes back and is stopping some of this positive feedback in that it's degrading an activated factor 8, which is part of the intrinsic factor, and it also breaks down an activated factor 5, which is part of the final common pathway, thereby stopping the progression so that we won't have our blood forming one huge blood clot. So it's putting a stop to that positive feedback. So even though you have never heard of protein C before, it is an important factor in keeping you healthy when you cut yourself and limiting the scope of the coagulation. And now there's one more compound, and that is known as antithrombin, which, as you would expect, is against thrombin. It's against forming blood clots. So at the same time protein C gets activated, all these other things are being activated. Antithrombin is also activated. And I'm not going to talk about how it's activated. But what it works on is not only against thrombin, to inactivate thrombin to limit the clot production, it also activates multiple clotting factors in the intrinsic pathway, as well as factor 10, which is the first one of the final common pathway. So this puts a limit on the further amount of coagulation. So what's the fate of a blood clot? Well, the clot gets formed. After the clot is formed and the platelets have stuck and we've got fibrin in there, the whole thing kind of contracts down on itself, and the fibrin particles are pushed more and more, and everything becomes very compact, and this is what's going to stop the blood loss. And actually, when this contraction occurs, because the platelet plug was actually stuck to the endothelial layers of the blood vessel wall, this pulls the cut edges of the damaged blood vessel together. This entire process of clot retracting takes about 30 minutes. And after 30 minutes, the clot is pretty stable. And at that point, we will start the factors that are going to break it down. For instance, our fibrinolysis system, where we will end up with those fibrin split products and release the serum, which can become part of the plasma again. But also realize before fibrinolysis comes into play, we also have those other systems, the antithrombin and the activated protein C, which are limiting the scope of this blood clot being formed. So take a moment, review the 3 basic steps put in the fibrinolysis, as well as the limiting factors against coagulation cascade. And here are all the little steps to make sure you kind of understand where in this. And remember, the protein C and the antithrombin, the fibrinolysis, that's all affecting only that 3rd step of coagulation cascade. It has no effect on the first 2 steps. So that is that, and I will see you again shortly. Thank you for all your hard work and making sure you understand this fascinating material.